If you're going to make people go through all of that or ask them to take that ketogenic diet, you kind of want to know that there's a good chance that they're going to respond to it, at least, down the line. So what the ultimate goal for us is, well, twofold. One is not just to understand the mechanisms for science sake, is to actually determine from that specific measures at the beginning of the trial, which will tell us whether that person is going to respond to the ketogenic diet.
An absolute powerhouse team of researchers is starting a new trial investigating bipolar disorder and ketogenic therapy as a potential treatment for it, learning the mechanisms at multiple different levels with brain imaging, with mood scores, with blood tests, looking at mitochondria, looking at mice and rats, and tying it all together about learning more about who may benefit most and all the different intricacies of treating mental health disorders with ketogenic therapy. Welcome to Metabolic Mind, a nonprofit initiative of Baszucki Group, transforming the study and treatment of mental disorders by exploring the connection between metabolism and brain health. Thank you for joining us on this journey. Before we jump into the interviews, please remember that this channel is for informational purposes only. We're not providing group or individual medical or healthcare advice or establishing a provider-patient relationship. Changing your lifestyle, trying ketogenic therapy, changing your medications, any of that without proper supervision can be very dangerous. So please always consult your clinician. And we realize that not everybody responds the same way to ketogenic therapy. There's a variety of different responses, and that's part of why this research is being done, so we can learn more. So with that, let's get on with the interview about this exciting new study with these wonderful researchers. Well, Dr. Mary Phillips, it's so great to have you here at Metabolic Mind to talk about your exciting new study. So thanks for joining me.
Sure, very happy to be here today talking with you, Brett. Thank you for inviting me.
Of course. Now, as we're going to get into, it's really a sort of a broad study with multiple different components to it. So why don't you start by just giving us an overview of what this study is and what you hope to sort of investigate and learn from it?
Yeah, sure. So the group of us, Anna, Colleen, and myself, and Zach, really very interested in understanding how the mechanisms by which the ketogenic diet works. And we're interested in understanding how it works at the sort of the wider, and also the wider brain scale, but also the smaller molecular scale level. The overarching goal really is to test the hypothesis that is becoming sort of increasingly known, but perhaps not so well studied. The hypothesis that what the ketogenic diet actually does is boost GABA, GABA being the inhibitory brain neurotransmitter or neuromodulator. And the reason that that's important is that not only does that sort of help reduce or improve the excitatory inhibitory balance in the brain between GABA and the glutamate, the excitatory transmission, but GABA is also a key modulator of dopamine. Now, dopamine is thought to be, hyperactive or produce too much in disorders like bipolar disorder and schizophrenia. So if GABA is modulating dopamine and sort of reducing the extent to which dopamine is produced too much, shall we say, then we feel that that's a way by which the keto diet may actually help people who have too much dopamine in their brains, i.e. if they have bipolar disorder or schizophrenia. So that's where we started. And what we're trying to do with this study is a number of things to test that hypothesis. One, which is my main study, is the human study where we're looking at brain imaging to test dopamine in the brain, but also GABA and glutamate, and various measures of mitochondrial metabolism, where the ketone bodies are actually working to restore the GABA, glutamate, and dopamine balance. And we're looking at various blood measures as well. And then we also have worked with Anna Andreatsu in Toronto in Canada, where she's working with us very closely, taking some blood samples from our patients and then creating cerebral organoids, and from them dopaminergic stem cells from people with bipolar disorder. So she can actually look at the cells, sort of the proxy measure of dopamine cells and their functioning from our participants with bipolar disorder. So at the same time as we are doing the larger scale brain imaging in people with bipolar disorder and healthy controls, she's also looking at their stem cells at the cellular level. And working with Zach Freiberg here in Pittsburgh, who, we also are going to get an understanding of the even more detailed structure of those dopamine cells in people with bipolar disorder. And then finally, we have Colleen McClung, who's doing some very elegant work in mice, also looking at the way the ketogenic diet impacts sort of dopamine function in the brain in mice who are bred to be a bit sort of bipolar disorder-like in terms of abnormal rhythms and hyperactivity. So overarching story is we want to test this hypothesis by going from mice to cells to circuits to behavior, using imaging and all sorts of other technologies in people with bipolar disorder. And
yeah, it sounds impressive. I mean, so many different modalities of it.
yeah,
Now,
yeah,
I want to circle back though and focus in on dopamine, because you brought up dopamine, and dopamine is sort of a hot topic in the popular media right now.
yeah,
People hear about it like from social media. It triggers dopamine, and people need to go on dopamine fast. So, tell us real briefly why dopamine. Yeah, obviously it's a natural component of our body. It's a natural chemical. We need it. It serves a
yeah.
purpose, but there can become a point where it becomes problematic. So tell us about, what happens when, in theory, when dopamine becomes problematic, what it can do.
Yeah, so as you quite rightly say, Brett, dopamine is critically important because it helps us process rewards, find things engaging, motivate. It's a very keen chemical in the brain that helps us become motivated to seek rewards and goals, etc., and drive. However, if there's too much, what happens is people go on almost like super drive, overdrive. And what is thought to be happening in people with bipolar disorder, at least, is that there's too much dopamine being transmitted into the key reward, the brain reward circuitry. So whereas it's good for you to have a certain amount of dopamine in order to, for example, getting out of bed in the morning or reaching a goal, working hard, that's very good. But if it's too much, it can make you almost take risks and become very risky in your behavior, sensation-seeking, impulsive. And then that's bad because obviously you get yourself, and people can get themselves into all sorts of difficult situations. So that's one way in which too much dopamine can be bad for you. Another way is that it can effectively cause all sorts of, movement problems, and you're hyperactive, not in control of your motor function too much. We see the opposite side of that in people with Parkinson's disease, where they don't have enough dopamine. And unfortunately, when they're given dopamine as treatment, it's not very well fine-tuned. So what happens is that suddenly they'll have all sorts of sporadic movements, which are not particularly pleasant, as well as this risk-taking behavior that I was just talking about. And so there's all sorts of problems that too much dopamine, can do and can produce. So it's not just risk-taking, it's not just sort of movement disorders, it's all sorts of sensory disturbances. So it's thought that, for example, that in schizophrenia, the hallucinations that people have might be in some way the result of too much dopamine affecting the sensory processing, as well as the motoric processing and the cognitive and the risk-taking behavior. So, it's almost like you need that sweet spot. If you have too little, you can't move. If you have too much, you're going to be taking risks. You're going to be having all sorts of abnormal movements. You need that dopamine to be completely fine-tuned in that sweet spot. Otherwise, there are problems at either end. And unfortunately, the medications that we all know about that, are produced and are given to people with these disorders, while they work to some extent quite well, they have the side effects because they too much of a blunt instrument. They're going to knock out the dopamine too much, or they're not going to stop the dopamine produced in the regions that you want the dopamine to be modulated. It's basically too much of a blunt instrument. So
yeah. So is the hypothesis then, with a ketogenic intervention, by balancing the GABA and the glutamate, you could potentially hit that sweet spot of dopamine, and that could be a mechanism for how it helps people with bipolar disorder?
Absolutely, because what we're doing is we're not taking a sledgehammer here. We are actually using body's natural resources to actually, beef up those natural resources a little bit more, so that rather than just blocking out dopamine or reducing it in a blunt way, we're actually modulating it in a more nuanced way by boosting this GABA. And just to say a little bit more about the GABA, the GABA is known to sort of interact with dopamine at many levels in the brain and in very many different places, but particularly in this reward circuitry that I was talking about earlier. So there are fine-tuned relationships between GABA neurons, GABA brain cells, and dopamine brain cells. And what we're trying to do is sort of boost the GABA component side of it. Now, I have to say, at the same time, what we think the ketogenic diet might be doing is by increasing mitochondrial efficiency. It will also help with dopamine metabolism, and within the cells that produce dopamine, it will effectively. We are predicting it will stop dopamine from being effectively produced too much at that level. So it's not just via GABA that we feel that the ketogenic diet might work. It might actually help with dopamine directly. But that's to be tested, and that's to be determined.
Right. So what's the protocol then? You mentioned people with bipolar disorder and healthy controls, and you mentioned a keto diet. So how does it all fit together?
Yeah, so it is an ambitious study, but we're very excited because we've already started the study. So we are going to be recruiting to test in full about 60 people who have bipolar disorder, or any type of bipolar disorder. And what we're trying to look at are people who are either currently euthymic, so they're not manic or they're not depressed, and people who are manic. But we are going to be also looking at people who also are depressed as well. But we're prioritizing looking at people who are manic and people who are currently euthymic. And then what we're going to ask them to do after a series of baseline assessments with their brain function, their brain scans, blood tests, etc. We're then going to ask them to start either the ketogenic diet or continue with their diet as normal. Now, obviously, as you more than any other person knows, it's not trivial to start a ketogenic diet. And some people find it easy, other people find it really hard. So we're allowing people to ramp up for about, 5 days to 3 weeks. And then once they're in ketosis, which we're going to be measuring daily with the Keto Measure app and machine that tests the blood. Once they're in ketosis, we'll officially be monitoring them on the diet. And then at the halfway through each phase, the diet is normal or the ketogenic diet, we're going to be testing their bloods again, just for all, not only for ketones and blood glucose, but for critical indices of metabolism, liver function, renal function, etc., insulin resistance. And then at the end of each 8-week period, we're going to test their bloods again and also do another brain scan, a series of brain scans. So effectively what people will have, they'll come in, they'll have their baseline assessments, they'll go onto the ketogenic diet after the ramp up for 8 weeks. Halfway through, they'll have the blood test. At the end of 8 weeks, they'll have more blood tests and another brain scan. And then they'll go onto the other diet, whether it's the keto or diet is normal, the same thing again. 4 weeks later, blood test. 8 weeks later, at the end of the whole study, they'll have the brain scans as well as the blood tests again. And that's it. It takes a village. I mean, obviously, we're very grateful to the Baszucki Group for their wonderful support. But, we have dietitians on the team, we have research associates, clinicians, neuroimaging data analysts, lots of people who are, very interested and also expert in other aspects of bipolar disorder. What I haven't said is that in addition to all of that, we're going to be monitoring people's daily activity using actigraph watches, and to determine how the ketogenic diet might kind of balance out, moderate, and start to normalize sort of abnormal excessive behaviors and risk-taking behaviors and just excessive movements that we see in people particularly when they're manic, when they're bipolar disorder. So we're looking at that too. So it's a lot of expertise, and very exciting.
So you've mentioned a couple times the brain imaging scan. Now, I assume this isn't just some scan you can walk into your local radiology department and get. This is a sort of a high-tech, advanced research scan. So because of that, is it an in-person only study? Do they have to be able to come into your facility to get the scan done?
Yes, at the moment, if we're going to actually ask people to do all components of the study, including the brain imaging, yes, it will have to be in person because the brain imaging has to be in person. Now, the brain imaging is 2 brain imaging sessions. One to look at the function in the brain, rewards and the dopamine. The other is to look at the GABA in the brain and the glutamate and lactate as a measure of mitochondrial function. So those will be in person. However, we'll see how the study goes. There are aspects of the study that could be done remotely. Obviously not the blood tests or the brain scans, but being on the diet itself and measuring, the actigraphy watch, and other questions that we have are all done, could all be done remotely once people come in for the first time to have their actigraph watch fitted, obviously. So some aspects of the study could be done remotely. If we were just interested in, clinical measures and the actigraphy, movement, etc., then sure, all of that could be done remotely after the actigraphy watch is fitted. But the brain imaging and the more detailed blood tests would necessitate coming in person. Yeah.
Yeah. And now when you're talking about GABA and glutamate, that's something that is sort of prevalent throughout the epilepsy literature about the effect of ketosis. So do you have any reason to suspect that it would be different in a population with bipolar disorder rather than with epilepsy?
This principle remains the same, that we're trying to boost GABA or normalize the balance between GABA and glutamate. The difference is where in the brain. And I think, the ketogenic diet probably, the reason it's so successful in epilepsy is it's impacting GABA, likely, it's thought, in various regions in the brain. Now, for people with bipolar disorder, it might be that only in certain places in the brain do we need to redress the balance, because we're looking particularly at the brain areas where there's too much dopamine produced. So in this case, we're looking at how GABA normalizes dopamine function, as opposed to how GABA just normalizes overexcited brain in general. So that's, I guess, the difference, but the principle remains the same. For schizophrenia, a lot of my colleagues are very interested in the ketogenic diet and schizophrenia. Again, similar to bipolar disorder, we're looking, more at the interplay between GABA and dopamine, as well as GABA on brain activity per se. So I think there are so many parallels, which again excited us to think that, the ketogenic diet there could be a real mechanism here, understandable mechanism related to the mechanism for epilepsy, GABAergic story.
Yeah. And, I think it's always interesting trying to balance this question of, does it work and how does it work? Yes. Sometimes two separate questions. And,
very much so.
And from a patient standpoint, they just want to know, does it work? But from maybe the clinician, the researcher, diving deeper, you want to know, how does it work? So tell us about, for you, how you see that balance in research and in clinical practice.
Brett, that's such an interesting question. It doesn't just apply to this study. It applies to, I think, virtually every research study that is involving, an intervention. The bottom line is that it's obviously the most important thing is you want to know if it works. But you see, the question then is, if it works, it might not work for everybody. And as I said earlier, the ketogenic diet is tough to get onto for some people, not everyone, for some people. And if you're going to make people go through all of that or ask them to take that ketogenic diet, you kind of want to know that there's a good chance that they're going to respond to it, at least, down the line. So what the ultimate goal for us is, well, twofold. One is not just to understand the mechanisms for science sake, is to actually determine from that specific measures at the beginning of the trial, which will tell us whether that person is going to respond to the ketogenic diet. So, for example, it might be that if we're right in our hypothesis, high levels of glutamate to GABA ratio at the beginning in the brain may be a clue telling us that it's those people who are going to respond best to the ketogenic diet. But it might be people who have really excessive amounts of dopamine and very low GABA who are going to respond very well to the diet, and other people might not. So that's the kind of thing that we really want to get out of this study. Ideally, having a marker that can be translated and scalable. And obviously, brain imaging has its limits, but we're hoping to have proxy measures of the brain imaging measures that you could do anywhere and say, well, you're likely to respond well to the ketogenic diet because we know how it works. And you've got the starting points telling us, you've got that mechanism that makes you bipolar. Therefore, the keto is the one for you, as opposed to lithium or something else. So that's really, the goal.
Yeah, it's such a crucial point of tying it back to how does this help the patient and help the doctor make sure they're treating the patient appropriately. So important. So I'm so glad you recognize that. Well, thank you. Thank you so much for taking the time to talk to us about your study. And I can't wait to have you back to talk about the results. So thank you again.
Look forward to that very much. Thank you, Brett, for having me. Thank you.
Well, Dr. Colleen McClung, thanks for joining me. We got to hear from Mary Phillips all about sort of the overview of the study and the different components. So now I'm excited to hear about your specific part of the study that you're leading. So please tell us sort of what is the general setup of this part of your study and what do you hope to accomplish with it?
Yeah, so thanks for having me. So We are doing the mouse component of this study. So, as Mary told you, we're really interested in trying to understand the ketogenic diet and how it relates to the treatment of bipolar disorder. And so, through using mice, we can do some experiments that we can't do in human subjects, obviously. So we can do more things in the brain. We can do some direct manipulations and see what happens to different circuits of the brain or gene expression and metabolic function, mitochondrial function, things like that. So our part of the study, we're going to be kind of paralleling what's happening with the human studies. So in the human studies, they're giving people with bipolar disorder a ketogenic diet and looking at some brain imaging and looking at some mitochondrial related markers and metabolic markers and markers of dopamine. Dopamine is a really important molecule in the brain that's thought to be important in bipolar disorder. So in our study, we were fortunate enough a long time ago now to identify some mice that have an increase in dopaminergic activity in the brain's reward system. And they also have a number of phenotypes, behavioral phenotypes that are very reminiscent of bipolar mania. So they're very hyperactive. They have increased exploratory drive. They have sort of a hyperhedonic phenotype. They find everything more rewarding. So, drugs, food, alcohol, everything. And if we give them drugs like lithium or valproic acid, we're able to reverse their behavioral phenotypes and reverse a lot of their hyperdopaminergic phenotypes. So they've been a really useful mouse model for us to study in terms of trying to understand the molecular mechanisms that underlie bipolar disorder and how potential treatments work. And so in this study, we're going to be giving the mice a ketogenic diet. So you can actually buy that for mice, and they'll be eating the diet, and then we can do again what, as I mentioned, a number of neuronal measures, molecular measures, metabolic measures on these mice to see what the diet is doing to the brain. So we'll be measuring dopaminergic activity in the brain, so doing some electrophysiology. We're also going to be doing some molecular studies to see what kind of gene expression changes occur in the brain in these dopaminergic regions in response to the diet. We're going to be doing some measures of mitochondrial function in the brain, and really just kind of getting a big picture of what the diet is doing to mice in the brain, and how we might use that to identify mechanisms as to how the diet is working.
Yeah.
And we'll be comparing it to some of our studies with mood stabilizing medications.
Yeah, I mean, it is interesting to think about all the things you can do in a rodent model or mouse model that you obviously can't do in a human model. But then there's, the pushback. Well, it's mice. Their brains are different. They, go
Sure.
into ketosis differently. The food is different. Like how do we directly compare it? So when all is said and done, how do you think your results will be able to, directly impact either further studies or further treatment for human beings with bipolar disorder?
Yeah, yeah. No, it's obviously mice are not people, so we do have to, take what we learn from mice and see if we can adapt that to human studies or use that knowledge to human studies. Fortunately, a lot of the systems that we're looking at, including these dopaminergic systems, are highly conserved between mice and humans. They're very old parts of the brain, old systems that are really involved in very fundamental processes to life, right? So reward seeking, eating, sleeping, these kinds of things. So there's a lot of conservation between mice and humans, and we already know that a lot of the mechanisms of action of some of these other medications that I talked about, so things like lithium, valproic acid, they do some similar things in mice that they do in humans, and so um, while mice are not humans, and we have to think about that, they're still a useful tool in terms of trying to get some basic mechanisms identified, and then of course the next step is then to test that in human cells and also in eventually in clinical models. levels.
Yeah. Yeah, I think that's important to sort of bring it back around, and I like how you're thinking about that. And well, it's exciting to think about all the different components of the study and how much we're going to learn from it. So, thank you for sharing sort of the overview of what the study entails. And like I said to Mary, I can't wait to have you back on to talk about the results sometimes in the future. So, thank you.
Yeah, thank you. We're excited about this too. Definitely.
Well, Dr. Anna Andreazza, thank you so much for joining me today here at Metabolic Mind.
Thank you for having me. It's a pleasure to be talking with you today about our study.
Yes, we've heard from Mary Phillips, we've heard from Colleen McClung. This is a very exciting study with lots of different elements. So now we get to hear about yours, which, we've gone from sort of like the human being and the brain imaging to the rodents and the rats. Now we're going down to the cells with yours. So we're getting like super small now. So tell us about, why you're studying mitochondria. Give us an overview of mitochondria and how this ties into the overall theme of this study of looking at mechanisms of bipolar disorder and ketogenic therapy.
Thank you, Brett. That's actually a very good point, because we should start to perhaps define what is mitochondria. So mitochondria is these tiny little cells inside of your cells that are your powerhouse of the cells. But they do a lot more than that. They control your inflammation response, they control your free radicals response, they control antioxidants response in the cells. So what we want to do with this study specifically is understand how the ketogenic diet is helping those little tiny mitochondria to actually breathe better, to do their job better within the cells. So to do that, we do in two ways. First, we look in the blood of the patients prior they started the diet, and then along on the continuum of the trial. And we measure in the blood biomarkers that can tell us how the mitochondria is responding to this diet and how the inflammation is responding to this diet. So that is a jitsi of a little bit of like the first part of the project. And then the second part of the project, we enter a little bit more in details in the cells, and we take your blood and we actually get one fraction of your blood called the white blood cells. And these white blood cells contain mitochondria. So we actually can transform them. It's like, think about like going backwards. We teach these white blood cells to go backwards in time and become stem cells. Once they become stem cells, which we call induced pluripotent stem cells, they have the capacity of transforming anything. In this specific project, we are interested in teaching these stem cells to become little brain organoids. But brain organoids that produces the dopaminergic neurons, because we want to understand whether the ketogenic diet is able to modulate the response in the brain of these patients on actually changing the levels of not only the mitochondrial response, not only the inflammation, but do they change the dopaminergic neurotransmission in this neurons. So that's a little overview. So I stop here and let you ask me other questions.
Yeah, so Mary Phillips talked about sort of brain imaging and how they can pick up dopamine levels with that. So measuring sort of, I guess you could say, on the macro level about dopamine. So now you're looking at the micro level of dopamine as well. And are the dopamine connected with the mitochondria then, or are those two different ways that you're looking at the cells?
Great questions, excellent questions. So in my lab, we produce the iPSC cells, the stem cells that will generate the brain organoids that have the genetics of the patient. And then we treat these organoids with ketones. We add, it's like you give in the ketogenic diet, but in a dish for these organoids, right? And then we put in the ketones inside them, and we see the response of the ketones changing the mitochondria or not. We'll see what happened there. And we would also measure the levels of dopamine in these organoids, which is measured by Dr. Zachariah Freiberg at University of Pittsburgh. The two indeed are connected. Mitochondria, when it becomes less functional, it produces oxidative stress, meaning it release free radicals. Imagine like free radicals as a troublemakers. They actually run around and they try to bounce in something, bounce in another thing, and they can actually bounce in parts of your proteins, your lipids, your DNA. And one of them is dopamine, and they can oxidize the dopamine. If they oxidize the dopamine actually don't act very well, and they become a stressor for the system as well. So yes, there is a connection, and we'll learn more as we go from here. And can the ketogenic diet, the ketones in this specific dish experiment, protect against that?
Yeah, and I gosh, I just think it's fascinating, the technology and the science behind how you're even doing this and creating these cells that become, brain cells basically, and measuring it. It's fascinating, and we could probably have a whole hour conversation just about the details of it. So I encourage anybody who's interested to look you up and follow more about what you're doing. But so then tying this back into the overall goal of understanding the mechanisms of bipolar disorder and how ketogenic diets can influence that. So what do you hope to take from this, and maybe think about what the potential next steps could be based on what you're going to find from your part of the study?
That's, again, a lot of fantastic questions. So to put everything in perspective, there are two things we know very well in patients with bipolar disorder. One, we know that the energetic system is dysfunctional in majority of the patients. One, is this in every patient? Probably not. So that's lesson number one we're going to learn in this trial. How many of those in the trial have a dysfunctional energetic system? Is that the dysfunction of the energetic system made them more benefited of the ketogenic diet or benefit less? So that's one of a big lesson that we learn, to move the field towards some bigger studies that can actually now understand who might benefit properly of using ketogenic diet. That's all. The lesson number two that I think we're going to learn is the inflammation. Inflammation, as you know, and many of us know, is key on the pathophysiology of bipolar disorder. And the ketogenic diet has known to improve mitochondrial health. By improving mitochondrial health, those troublemakers, the free radicals, they calm down, and they do not activate a protein called NLRP3 inflammasome, which leads to a cascade of inflammation. So we will be measuring specifically this pathway. So again, it's possible for us to understand, one, is this ketogenic diet ameliorating, improving this pathway of inflammation in these patients? And if it is all the patients in the trial that benefit, or a portion, or a big portion, a small portion? So those are lessons that we still don't know, but it will be very valuable for the future studies.
Yeah, I think that's so important. And Colleen and Mary gave us similar answers about how everything they're doing ties back to just learning more about who are the appropriate people, who's going to benefit most. And I think that's so important to bring it back to how we can help people and help people better. So I'm so excited about this trial, and I hope we can meet again and talk about the results. And thank you so much for joining me to explain your portion of it.
It was a fantastic time to be here, and I hope to talk more when the results are there.